Hello, everyone. My name is Fernanda. I graduated from vet school at the Federal University of Pampa in Brazil and at Santa Cruz State University, also in Brazil. I did my master's and I'm currently doing my PhD focused on the study of the Maine sloth's health. I'm also a research associate for Instituto Tamanduá, the Institute for Research and Conservation of Sloths and Eaters and Armadillos in Brazil. So before starting the presentation, I would like to thank Kenny for the invite to be here today. It's just such a pleasure to share my experience with you all. So let's start it. So Maine sloth, 3 toes and 3 challenges facing the study of their health. Right. There you go. So there are 2 types of sloths. The 2-toed sloths that have 2 toes in the front limbs, like you can see here in the picture. And they are represented by 2 species, the Coloepus didactylus and Coloepus hoffmanni. They are found in multiple countries and they can have different common names, like I'm showing you guys in the slide. And we do have the 3-toed sloths that have 3 toes in the front limbs, and they are represented by the species Bradypus variegatus, Bradypus tridactylus, Bradypus pygmaeus, and Bradypus torquatus. And more recently discovered the Bradypus crinitus. So some of the different species, they can be found in multiple countries, like you can see here, and others are found only in 1 country, like those 2. And the star of the show today is the Maine sloth. So we'll focus on the species that are only found in Brazil. So until 2022, we only knew of 1 species of Maine sloth, the species that we call Bradypus torquatus. But in 2022, a taxonomic review was published and indicated a new species of Maine sloth, like I mentioned, the Bradypus crinitus. This review was based on morphological analysis, field observations, DNA analysis, and they demonstrated that actually we have 2 species of sloth that can be recognized, the Bradypus torquatus and the Bradypus crinitus. So the Maine sloth is the only sloth threatened by extinction in Brazil and is classified as vulnerable B2A, B2-3. I just want to make sure that when we say that the Maine sloth is in the vulnerable category, we are talking about the species Bradypus torquatus, because until now, no new risk assessment has been carried out to include the other species, the new one, so the Bradypus crinitus. So the classification vulnerable B2, AB to 3 means that the Maine sloth faces a high risk of extinction, in nature, that the fragmented area is estimated at less than 2,000 square kilometers. This area has high fragmentation and continued decline in the occupation area and also in the extension area. So if the Maine sloth, the Bradypus torquatus, already had a limited geographic distribution, already had a low population density, and was already threatened by extinction, so what about now that we are finding out about a new species? So a new risk assessment for the Maine sloth is urgently needed now for both species, the Bradypus torquatus and the new one, Bradypus crinitus. So let me press OK here. So we can visualize all that I was talking about better with this map. So the distribution of the Maine sloth in Brazil is restricted to the Atlantic forest, and it's confirmed in 4 states and not confirmed in 2. And where it says Bradypus crinitus now, before it was considered Bradypus torquatus. So now the Bradypus torquatus is considered the species that are in the northeast of the country. So now we say northern Maine sloth and southern Maine sloth. So for the ones in the south, the new species, southern, and the other ones in the northeast of the country, we say northern. And the name of the state, so I'm gonna mention a lot, I'm just gonna mention here, so Sergipe, Bahia for Bradypus torquatus, Espírito Santo, and Rio de Janeiro for the southern. All right. So what are the threats, right? The threats facing the Maine sloths, they are subsistence hunting, illegal trade, wildlife equal collision. And here I bring an example of wildlife crossing in one of the highways in the state of Bahia. So the state where we can find Bradypus torquatus Maine sloths. And of course, the main threat is deforestation. The Atlantic forest has been overexploited with for logging, charcoal production, and cattle pastures. And the Atlantic forest is one of the world's biodiversity hotspots and has already lost 72% of its area. It has been reduced to several small fragments. So here in this picture is a great example of an area in the south of Sergipe, one of the states that we can find the Bradypus torquatus. And it's one of the last preserved fragments of the Atlantic forest in the state. So Instituto Tamanduá, the Institute for Research and Conservation of Sloths, Armadillos, and Anteaters in Brazil, they did a scientific expedition in that area to capture and collect samples from the Maine sloths there for the first time. And it was very nice. National Geographic covered the expedition. All right, so like I said, the Atlantic forest is being reduced to several small fragments, and it's mainly due to predatory hunting, illegal logging, invasion of exotic species, and also parasitic diseases. And we know that the Maine sloth is restricted arboreal. So spending much of its time, most of the time, in the tree canopy. So they move slowly between branches, and they rarely come down to the forest floor unless if they need to defecate. So considering the extensive deforestation and fragmentation of the area, this behavior of them descending to the forest floor may also be related to the lack of connection between trees. We just mentioned deforestation. So if there are no trees connecting, they need to go to the ground to go to the next tree. So since the infection by different parasitic agents, they can occur through contact with soil and vegetation shared by species, or even contact with other animals, other wild animals, this behavior of descending to the forest floor can be a deciding factor for the occurrence of an infection. And the presence of humans, domestic animals, which more and more we see, they are moving closer to the natural reserves. So that increases the possibility of spreading parasitic agents to new environments and to wild species. So although the parasitic agents play an important role in maintaining ecological and ecosystem diversity, they can also threaten biodiversity. They can lead to a decline in natural populations, reducing the survival or even the reproduction of the host. And they can also be responsible for local extinctions of endemic species, and it was already reported before in other studies with other wild species. So we need to have that in mind. It's very important to study parasites anyway, especially in the species that are threatened by extinction. And there is very little data and health studies on the Maine sloths populations. Well, so let's start with the challenges of studying the sloth's health. I'm bringing then 3 challenges today. Number 1 challenge, finding the Maine sloths. So I brought this picture today because I feel like most people have this expectation seeing the sloth that close in the forest. So when I tell people I'm going to do field work and look for Maine sloths, they don't usually think about needing binoculars or a neck massage because you do look up all the time. So I'm going to bring the reality to you. So I'll give you a few seconds to look for the Maine sloth here in the picture. There is like a view from the forest floor and a view from a zoom. I zoomed in. I feel like it's almost a where is Waldo challenge. Waldo, Wally, depending on where you grew up in the world. In Brazil, we say Wally, but I know that in the United States, we say Waldo. So where is Wally? Where is Waldo? All right, so now I'm going to review. So here in the middle of the circle, you can see the Maine sloth. So here I'm zooming in even more with the video so you can take a better look, and you could easily be mistaken for dry leaves, shade. So it's hard. But now level hard. And let's consider that this is a picture through binoculars. So imagine having to spot a sloth there from the forest floor. It's very difficult. So I'm just gonna show you guys where the sloth is here. So it makes me just amazed at how great they are at camouflage. This picture is just the proof that you need experienced people on your team. There is no way you can tell that there is a Maine sloth here that's like hair. If it's your first rodeo, you just won't. It's very difficult. It's very tricky. So, and here is a picture of a few people from an exceptional team from Instituto Tamanduá. They're looking for the Maine sloths. And it's the great example of the need of the neck massage that I mentioned, because you just look up for hours looking for the sloths. It's very intense. All right, challenge 2. So capturing the individuals. This is a professional climber of the team, one of the few we can actually find in Brazil that works exclusively climbing and capturing different species in Brazil. He has a lot of experience with Maine sloths and capturing many other wildlife species. And he knows different techniques, and each situation will demand a different technique. So all that because the sloth needs to be captured safely, but the climber needs to feel safe as well. It's just not the animal, it's both. So here, for example, the Maine sloth was in a branch that he could not have safe access to. So using this slingshot, he can get to the branch where the sloth is, he can shake it, and then lead the sloth to move to another branch, move to a location that we could actually have access to, or at least a safe access to for both, for the climber and for the animal. And here is another example of a different technique. It's important to know how to work with the species, because when you get to the tree canopy, you you have to physically restrain the individual without hurting yourself and also keeping the animal safe. So when you were up there, you hand catch the animal, you wrap up the claws with Velcro or vet wrap, and then you put it in the cotton bag, and then you lower to the forest floor using this rope that was in the second video. And I'm saying that we have to have experience when you're up there to catch those claws and wrap it, because look at how dangerous it could be to be hurt with these claws. So when the main sloth want to defend themselves, they can spread their toes like that, as you can see in this video. And in this video specifically, the sloth is recovering from anesthesia, and it was already spreading the toes like that. So it wasn't its normal self, but was already in defense position. So they can open toes even wider, and it's dangerous, and it can actually hurt you pretty bad. So as soon as the individual safely gets to the forest floor, the vets step in to anesthetize so we can start handling the animal and collecting samples and all that in an effort to reduce stress to the animal, of course. And the anesthesia step is to ensure a safe operating environment for both animals and researchers. So you're already heard me before, it's also safety of the people and safety of the animal. And other studies with other species of sloths, they revealed that anesthetized individuals can present high blood pressure, different heart rate changes. So all that because of the proximity to humans. So they sometimes don't change their behavior. So you don't know necessarily, but they are changing heart rate and blood pressure. So earlier this year, we published the first report of chemical immobilization and physiological parameters for free-ranging Bradypus torquatus. So sloths from Bahia, from northeast of the country. And we used ketamine and medetomidine. It was a study with 12 main sloths, and it just indicated that this is a safe and effective choice for anesthesia of main sloths. So when you have this rare animal in front of you, you collect all possible samples. Even if it's not for your project, you collect materials for other researchers or even future researchers that you don't even know yet, but they can become interested in studying main sloth. Instituto Tamanduá has a great sample bank, for example. So when we are there in the field work, we collect hair, anal and oral swabs, body measurements as well. We collect blood for different tubes, so with and without anticoagulant, so purple and red tops. And we also collect ectoparasites. Here in this video, you can see ticks, but we can also collect moths. It's very common to see in sloths. And during this process of sampling, there is always a vet monitoring the anesthesia and recording the parameters. So temperature, heart rate, respiratory rate, and that's done every 10 minutes. So after we are done collecting everything, we administer the anesthetic antagonist. And after the individual is fully recovered, we rewrap cloths like it's showing in the video. But that's done when we can, because when we can't, we pinch the claws like it's shown in the video. And we release them at the nearest tree or even in the vines that they were where they were found, or even where they were actually found, the same tree. And we always release legs first and arms last, because they use the arms for defense. So doing it that way, you just can step back on time to be safe, right? All right, so let's move on to challenge number 3. So processing the samples. As you can see here in this picture, we are in the back of a truck. So we collect the samples in the back of the truck, but not always. Sometimes we just open a tarp on the forest floor, and if it's too far from where the truck is parked, we do it everything like on top of the tarp in the middle of the forest. So after we collect all the samples, we have everything with us. Sometimes we even have to hike back to the car. So we put everything in the backpack and then we head back to the car. But blood smears, we do it right away. So we got the sample, we do blood smears, and then we have a place to put it safely where it's not going to break, put it in the backpack, and then you can go down if you're hiking or you walk back to the car. But then what now, right? What do you do with the samples? Where are you? Is it close to where you're supposed to take the samples? So here I took a screenshot of the map from the reserve in Bahia, so where we work with Bradypus torquatus. And as you can see, the distance between the lab that we usually process our samples is about 7 hours by car. So we don't wait that long to process most of the samples. We can't risk losing such rare samples. We have to keep in mind we are working with a rare animal. It was like this super huge opportunity to find those animals that are threatened by extinction, and all that to lose samples. So we have to start processing the samples we can. So we create our own lab structure nearby. And when I say nearby, I mean could be a camping tent, could be a hotel room. As long as we have electricity or a generator, we can make it work, but it is improvised. Like here, super improvised. We do have partnerships with other universities and labs, but Brazil is just huge. It's so big. And the areas where the sloths live are usually very remote and very far from all the other universities and labs that we could have access to. So we have to do it in an improvised lab. So for our hematological analysis, for example, we have the equipment and we have the solutions. So total leukocyte count, red blood cell count, differential leukocyte count, hematocrit, total solids, all that we can do it right after field work in our structure built nearby. And for the biochemical analysis, we do leave the blood samples at rest for about 6 hours until we spin to obtain the serum, because we noticed that the clot forms better if we just wait the 6 hours. And we have ice, we have containers. So finally, to transport the samples the next day, we'll have all the equipment necessary as well. And we keep them refrigerated at 5 degrees Celsius. And not all of the samples will need to be kept refrigerated, right? Like ectoparasites, hair, we don't need to keep it in the fridge or anything. So that's easier to transport anyway. So when we finally get to the lab, all the facilities and equipment will be there for us, and then we can start processing the samples. So I specifically work with hemopathogens, so parasites in the blood. So when I get there, I get to extract DNA from the samples, do PCR, ELISA, and etc., right? All right, so I decided to bring a challenge. So if you are here with me, we'll have this bonus edition. And no kidding. allergy reactions, bug bites are a real thing, and it is a big challenge during a field work while you're trying to work with sloths and capture the sloth, get the samples. And sometimes that happens on day one of your expedition, and you have to go through it itching. No other word for that. You go through it very itchy. But it's very important to drink water. Dehydration is also a thing. It could be also a challenge if you are dehydrated. And even though I said that we need a neck massage because we look up all the time, we do have to look around because wasps, bees, bugs in general, snakes, they are there. So you have to pay attention to the surroundings as well. All right. So that was a video of different bug bites. All right. So if we combine everything that we talked about, and we talk a little bit about Maine sloth health as well, we'll wrap up this presentation, okay? So the health assessment of free-ranging wild animals is just essential to define the health status of a population. It will help you detect environmental issues, detect diseases, and even identify the risk of a population decline. And the data obtained from this assessment can be interpreted at both individual and population levels and can be very valuable for us to keep track of the health of the threatened species. So it's not going, sorry. There you go. So hematological and biochemical analysis, they have already been reported as the most common health assessment method in wild animals. So it is very important to evaluate the condition of an individual or of a population, like I mentioned before, and it indicates diseases, nutritional status, habitat quality even, so many different options. There are articles available related to hematological and biochemical values of other species of sloth, but related to the Bradypus torquatus, so the sloth that we find in northeast of Brazil in Sergipe or Bahia, there is only one study about it. And these hematological values, they are mixing captivity and free ranging. So I feel like it's important to mention that we don't have sloths, Maine sloths, pardon me, the Maine sloths specifically, we don't find them in captivity. For this study specifically, they were in captivity, but we don't have anymore. So it's hard to compare sometimes values from animal that is living in captivity and the animal that is in the wild for many factors. Diet can interfere, many other things can interfere, so we have to be careful. And there's no study with biochemical values of Bradypus torquatus. And there is no study at all with biochemical values or hematological values for the new species recognized, so the Bradypus crinitus. So now in my PhD, I'm working with a team of researchers to release the data we have been working on about the first hematological and biochemical values for the new species recognized, the Bradypus crinitus. So soon it will be available and can be very helpful. All right, so for my master's, I worked with hemopathogens in Bradypus torquatus, so parasites in the blood of the Maine sloths that live in the northeast of the country. I had samples from 29 clinically healthy Maine sloths. They, when I processed all the samples for all these hemoparasites listed here in the slide, they were all negative for Anaplasma, Babesia, Ehrlichia, Mycoplasma, and Rickettsia. But 11 individuals were positive for hepatozoon. And since all the individuals were considerably healthy, like I said, the detection of the hepatozoon could mean that the main sloth is just a carrier, has a role of a carrier. But we would need more studies to address this possibility. And there are other studies with other species of sloths that address this possibility, but with main sloth, there are none. So we have to investigate better. And it's important to say as well that the possibility of the main sloth developing clinical signs cannot be ruled out, because after we released the animal, we didn't monitor. So maybe they developed some clinical signs after, maybe they didn't. We just don't know. So once again, we would need more studies with the main sloths to have a statement like that. But if we consider the biological cycle of the hepatozoon, the hemoparasite, the only arthropod that can act as a definitive host that was found parasitizing the sloths the day that I captured was the Amblyomma varium tick. It's a tick that is also known as the sloth's giant tick. And this just indicates that transmission probably occurred through the Amblyomma varium tick, but maybe I just didn't see the day that I capture other parasites. So it's hard to say, but we did see the Amblyomma varium, the sloth giant tick. So we need more studies once again to investigate deeper. And it's important to say that this is the first study investigating the presence of any hemoparasite in main sloth. So we don't have other studies to go look to. And even though all the samples were negative for the other hemoparasites that I mentioned, we have to consider that the area where the main sloths are, they have a high rate of anthropization and has a lot of domestic animals. And when I say a lot, I mean a lot. So it's like on average 50 houses with about 2 dogs per house. And if we do the math very quickly, it's going to be at least 100 dogs in the area. And some of the families, they have other animals besides dogs, like horses, chickens, pigs, and it's out there then. So maybe they're not infected yet with many of the hemoparasites, but since they live very close to humans, very close to all these domestic animals, and they share vegetation with other species as well, the possibility is out there. So we have to keep that in mind. So right now in my PhD, I'm still working with main sloths and hemoparasites. But besides the species Bradypus torquatus, I'm working with the species Bradypus crinitus. So species from Sergipe and Bahia. So northeast of the country, Bradypus torquatus. And Rio de Janeiro, it's Bradypus crinitus, the new species. So I'm studying hemoparasites in all 2 species, and I've been getting very interesting results of positivity for trypanosoma and for toxoplasma. Soon we'll be publishing some new data as well, very interesting, so keep an eye for it. In 2022, during my master's, we published this scientific article about the sloth giant tick. So I have a video here of them, so you can take a better look. And it was a total of 36 individuals that were parasitized by ticks of this species. And it was like 50% of the amount of individuals we captured, they had the amblyomma variant tick. So this study confirmed the importance of the species Bradypus torquatus as a host for adults and nymphs of the sloth's giant tick. And I feel like it's important to mention that by then we considered all the main sloths in Brazil, Bradypus torquatus, and actually it's Bradypus crinitus and Bradypus torquatus. Now we know that actually all the two species, they are infected by the tick. Well, we can't talk about health and not talk about diet. I feel like sometimes people forget that animals' eating habits are indeed a much important health topic. When I tell people that I work, that I study Maine sloth's health, I ask them, what do you think I work with? And the answer is always disease. So a good, healthy environment is so much important for the animal's health. So anyway, this here, it's the same plant, the same tree, and it's a very common tree in Brazil, and we know this tree as the sloth tree. I know as Embaúba, the name of the tree, but there are different names for it, even in Brazil. Some people call different names, and I put the name in English here as well to help guide who is watching. And in fact, it is a common plant that the sloths eat, but it is not the only one. And sometimes people think when they are trying to rehab a sloth, they always go for this tree, they always go look for this plant specifically. So I'm bringing that up because this very interesting study identified 67 species consumed by the Maine sloth. And why are these results so important? Just for us to know, when we are working with health and we are working with animal conservation, we need to think about feeding protocols when they are in captivity. Sometimes they cannot have access to one plant, but they can have access to another. And in this article specifically, they track which ones were preferable by the sloth. So it's interesting to know which ones they like better. And it wasn't in all cases that they preferred this plant specifically. So maybe we were gonna try to feed this plant in captivity if we had it for rehab or something, and they would eat. So knowing that we have 66 more options, it's just amazing. And not only thinking about captivity, but also thinking about ecological reforestation. It's like just so important. If you want to work with conservation with the species, you also have to think about the environment. Deforestation is one of the biggest threats. So if you want restoration initiatives focusing on the conservation of the Maine sloth, that's great to know that there are 67 species of plants that they are consuming. So this is another interesting article that combines climatic factors and behavioral aspects. And the authors, they evaluate the influence of hourly ambient temperature variation on the Bradypus torquatus activities, postures, and tree crown positions, like we can see here. And they do that under sunny and cloudy conditions. And they tested if any changes to the animal's posture, and position increase their exposure to human detection. So it's very interesting. It's worth reading it. And we know the authors, and they mentioned that, but they were also talking to us about them noticing that the Maine sloths there, they were spending a lot of time and a lot of energy is scratching themselves. They were spending more time doing that than what other authors that actually study their behavior had found and what it said that it's normal for the species. So knowing that they are being observed and they are scratching themselves more, we can clearly make a connection to our current study. And let me see here. Look what we have been finding in the sloths. So here we are finding some scabs, some lesions. We find some alopecia in some of the sloths that we've been capturing. And we have been doing skin scrapes and skin cytology of those affected areas. And that's what we have been seeing under the microscope. So the species of mite identified is called Lobalgia strusardi. And it was a species previously reported to cause mange in other sloth species. So we are working on an article about this topic right now, and soon it will be available as well. All right, thank you so much for sticking around with me. I enjoy so much talking about this species. It was just such a pleasure to be here. Once again, thank you, Kenny, for the invite, and I'm open here to answer some questions.
Thank you so much.
Yeah.
Before we started recording, I mentioned that they're my favorite. And
yes.
Despite all of your bug bites, I would still like to come and visit you in the Wyatt Sloth.
Yes.
If anyone has questions, they can type them into the chat box and I will read them in a moment. But I just wanted to tell everyone about the specialist group very quickly. So this is the International Union of Conservation of Nature Species Survival Commission, Xenarthran Specialist Group. And our website is xenarthrans.org. We are active on Instagram and Facebook for about 30 days. We are asking people to give us their common names of all 38 Xenarthran species. We're also active on YouTube, where we post all of our past webinars. And we have some animated videos, which we'll talk about in a minute. When you go to our website, you will see that we have species profiles and also taxonomic information. And on our website, we have a lot of good scientific information for adults, including our journal, Xenarthra. And it was just accepted into the directory of open access journal, the DOAJ, which will allow our journals Xenarthra to reach a wider audience, which we're very excited about. And like I was mentioning on the website, we have scientific statistical information for adults for all the species. But in the past year, we have added lots of stuff for children. We have 4 animated videos, one on anteaters, one on sloths, one on armadillos, and then one on past, present, and the future of Xenarthrans. And all of our videos, all of our educational materials are available in Portuguese, Spanish, and English. To accompany the videos, we have coloring sheets and word searches and spot the differences that are all free to download. And we would like to remind everyone about some important dates that are coming up this year. On August 13th, every year it is International Armadillo Day. This year, International Sloth Day is October 19th. The date changes every year. It's always the 3rd Saturday in October. And World Anteater Day is always November 19th. It was changed many, many years ago, but from now on, it is always November 19th. And you can celebrate these wonderful animals by posting about them and sharing our posts. We like to do a webinar at least once a month. We have a couple of webinars coming up. The next one is in a couple of weeks, and it's about restoring area or rehabbing the tamandua in Mexico. And if you work with Xenarthrans in zoos or in rehab facilities or in the wild, and you would like to share your photos and videos with us, or if you like to share your scientific papers or graphics that you have in your scientific papers, you can give us a contact and we will happily share them. Because tomorrow we're going to be sharing some scientific news about 9-banded armadillos. If you feel inspired to help the conservation of Xenarthrans, you can go to our website and we have a little donate button and it tells you where your funds would be going. They can help our education and our conservation initiatives. If you don't want to just give us money, but if you want to get something in return, you can go to our spring store. And Fernanda, I am wearing this for you. I'm wearing my maned sloth t-shirt.
Amazing. Love it.
Now, I cannot tell you. We have one that we call the northern maned sloth, and then we have one that we call the southern maned sloth, but it's really the same cartoon, but just flipped. But we have armadillos, we have anteaters, and if you purchase one of our Xenarthran bling, we encourage you to send us a picture so more people can know about the wonderful Xenarthrans. So we have stickers, tote bags, tank tops, t-shirts. We got everything. And we always like to thank our partner institutions, FIA, the Foundation for International Aid for Animals, and Nurtured by Nature, because they help our webinars keep going and our educational programs. All right, so if anyone has any questions, you can maybe speak up. And I have two questions for you. Can you give us the symbiote load of an average maned sloth? Like, do they have 10 ticks and moths per sloth, or 50 or 100? Or with the mites that are so tiny, can you quantify the good?
It really depends. Sometimes we find 2 and sometimes we find 20, but I would say let's put it in the middle, maybe 10, but sometimes it's more. We do see a lot of them parasitized, so.
And the moths that we're saying are like commensals. They're not hurting the sloth.
Yes, exactly.
And can you give us like a specific instance of what type of invasive species are directly affecting the maned sloths?
So we have the Calitrix, I think that's how you say, like the monkey. I don't know the name in English, I'm sorry, but that we call it sagui. And actually the type of trypanosoma we've been finding right now, they are seeing in those monkeys that are not supposed to be there. They're invasive species. So we could maybe put a relationship between that, but we are studying it and investigating. But so far we are going on that track because we are seeing the type that we see in that monkey in the sloth. So maybe there is something there. So
where are the monkeys native to?
Australia, a lot of them. Yep.
Marielle, you have further questions or congratulations?
No, I'm just impressed. I mean, I'm used to working in the desert where you look down and search for armadillos, so it must be quite challenging just to look up all the time. And I love that picture where I really tried hard to spot the sloth and I couldn't. Not even when you put the arrows on it, I still couldn't see it. So
it's still hard.
Yeah, I guess I wouldn't be a good field assistant for that project. Definitely not. So it's amazing what you're doing. Congratulations. And it's so important because we just don't know enough about these animals. And as they are threatened with extinction, studying their diseases is even more important because, a lot of times we don't think about diseases being causative agents for extinction. So I think that's one of these huge unexplored areas that we need to take care of. So congratulations. I really loved it. Thank
Oh,
you.
thank you. Yeah, thank you.
Flavia, would you like to say anything?
No, only congratulations. Fernanda is a very, very good student, and she spent a lot of time with this in this project, you know. Obrigada, Fernanda, foi lindo.
Thank
Orgulhosa.
you. Muito obrigada, Flávia.
Wonderful. Any other questions?
Don't think so. I didn't get any more.
Okay, well, thank you so much again, Fernanda, and congratulations on your amazing work.
Thank you so much. It was a pleasure. Bye.
Bye.
Bye, take care.